Tasmanian devil facial tumour disease is a transmissible cancer that affects 90% of the Tasmanian devil population. Deacon, Bender and Pearce described this cancer as a colonial cell line transmitted by allograft. The term colonial cell line describes the origin of the cancer cell and how this entire disease originated from a singular mutation in one female devil. Allograft refers to the cancer and how it is spread by the devils biting each other's faces during mating and feeding. However, it is interesting to note that it is not the cancer itself that is responsible for so many deaths. The facial tumours grow so large that the devil will die from suffocation or starvation. Over the past 2 decades, scientists have been looking at the devil's genome to try and investigate the nature of this cancer and the type of mutations that occur. Through extensive research, scientists have used various gene sequencing technologies to compare the karyotype of a healthy devil to that of an affected devil, and find out how this disease escapes the devil's immune system. These technologies have enabled scientists to investigate how the tumour evolves on a genetic level and improve the chance of eradicating the disease. The main types of gene sequencing used in the investigation of DFTD is chromosome painting and G-band analysis. While both technologies give similar results, it is important to look at both as they contain individual discrepancies and inaccuracies, with the main issue lying within the procedures. The full protocol for chromosome painting is much more time-consuming and complicated than G-banding technology. Chromosome painting requires at least 10 different chemicals and different forms of equipment. After lots of pre-staining preparation, the chromosomes are taken from the cell during metaphase and stained with a fluorescent dye. This dye reveals light and dark areas of the chromosomes, and thus, the locus of each gene can be more clearly identified. This allows scientists to better locate translocations, insertions, and deletions. Recent developments of the technology have decreased the amount of background illumination from the dye, thus giving more accurate results. It is for this reason chromosome painting is more often used to identify incredibly complex chromosome abnormalities, and as seen in the case of Tasmanian devils, genomes with multiple chromosome mutations. Although similar to chromosome painting, G-banding provides slightly differing results. G-banding not only reveals the abnormalities in the devil's genome, but can classify the tumour suppressor genes. From here, scientists can localise these genes and eventually figure out a way to activate these genes in a living devil. However, G-banding does provide some limitations. Some studies found that this method revealed far less chromosome abnormalities than other gene sequencing techniques such as chromosome painting. While this gene sequencing technology cannot cure DFTD, it is incredibly helpful when monitoring the progress of tumours during different experiments. A study conducted by Deacon, Bender and Pearce used gene mapping and chromosome painting to compare the genome of a healthy devil to a genome of a devil suffering from DFTD. The study concluded, by anchoring genes to a reference and tumour maps, we can predict the locations of common tumour suppressor genes and oncogenes. By locating the suppressor genes, scientists have a better chance of changing the phonetic expression within the animal's genome or creating a vaccine against the cancer. Scientists both inside and outside DFTD field have gravitated towards using chromosome painting as a screening test for more specific complex cancers, as opposed to G-banding. In diseases such as chronic myeloid leukemia, it was found that chromosome painting revealed many more abnormalities than G-banding technology, and while the study was completed 20 years ago, scientists still believe that chromosome painting provides more accurate details. While this might be the case, both technologies are extremely useful throughout experimental drug trials and attempts to activate tumour suppressor genes. This technology is constantly under review and has been improved and modified to not only assist in the conservation of Tasmanian devils, but also extending as far as human cancer research. There's also been investigation into how this tumour evades the immune system of the Tasmanian devil. Pye, Woods and Kress explain that the tumour's constant adaptability and secretion of immunosuppressive cytokines have allowed the tumour to go undetected by the immune system. More advanced and specific gene sequencing technology reveal the nitrogenous bases within the transcriptome, and thus scientists can better understand the mutations in the mRNA. Not all chromosomal mutations are translated into proteins. However, cytokinetic proteins develop to become the leading cause in the devil's immunodeficiency and increased tissue necrosis. While many efforts have been made to solve the problem of DFTD, the methods used do raise some ethical dilemmas, as does any experiment that works with animals. Most studies completed have a variety of variables for the results to maintain accuracy. However, this does mean that some devils are intentionally left untreated or even purposely infected with the disease to create a more controlled testing environment. While many argue that this is for the good of the colony, it does raise the question of where do scientists draw the line between research and unnecessary damage? This is especially prevalent in recent studies as scientists are worried about the number of healthy devils left in the wild, with some studies capturing and collecting samples from as many as 69 wild devils per trial. To combat these concerns, many ethics committees, such as the University of Tasmania Animal Ethics Committee, in conjunction with the Department of Primary Industries, Parks, Water and Environment of Tasmania, have restricted the number of devils available. The study completed by the University of Tasmania were restricted to 9 maturely aged healthy devils for their research into a vaccine against the disease. There were also many strict protocols followed during the experiment that comply with the Australian Code for the Care and Use of Animals for Scientific Purposes legislation. To minimise distress and maximise the well-being of the animal in the given circumstances. The code states that unless there is evidence to the contrary, it must be assumed that the procedures and conditions that will cause pain and distress in humans cause pain and distress in animals. Decisions regarding the possible impact of procedures or conditions on the animal's well-being must be made into consideration of an animal's capacity to experience pain and distress. Thus, all procedures in the experiment completed by the University of Tasmania, such as biopsies and blood samples from the jugular vein, were performed under anaesthetic in order to minimise pain and distress. The code further outlines whom should be present in the experiment to oversee the care and treatment of animals. By following these protocols, the risks of unnecessary pain and distress can be limited. The creation of a vaccine and the investigation of tumour suppressor genes have been made possible by the countless gene sequencing studies completed over the past 2 decades. These studies aim to find chromosome abnormalities and locate tumour suppressor genes, and have become increasingly more successful as the technology has been developed. While some are concerned with the ethical issues raised when testing on animals, many protocols by various government and ethical committees have been put in place to address these concerns and improve the well-being of the animals. This technology has proven to be a reliable screening method and will continue to help unravel the mysteries of not only this disease but countless other cancers in hopes of eventually finding a cure.